Okay, so let's get started. Let's get started with this paper, regulation of ABCA1 expression by microRNA33. So this is the paper. I realized earlier today that I forgot to announce yesterday that I had posted them in Canvas. It probably won't make a huge difference if you read these papers in advance or not. In fact, there is one way in which is actually better if you don't, because then you come with an open mind and that can make for more fun conversations. But generally speaking, my plan is to publish the papers that we'll discuss in any given week, at least the Friday before or so. So today we'll talk about this paper, and the paper that we'll discuss on Wednesday is already posted in Canvas if you wanted to take a look at it. And when you do look at these papers, I would also recommend that in the interest of your time, not to engage with them all that deeply. If you would like to engage with the papers in preparation for class, one thing that I might advise is that you read the introduction and the discussion. And then you kind of skip all of the nitty-gritty, sometimes sluggish details about methodology and results, because those are the ones that can get a little time consuming. You may find yourself second guessing every term, and it's just makes things a little slower. But it's a good exercise when you're first engaging with a paper. Just go through the introduction and the discussion. That will give you some storytelling, some narrative about the story, and then you can go back and, if you want, engage with the results much more deeply, which is what we'll do here anyway. So this is the one that we'll discuss today, and I wanted to go through the introduction, the main points, briefly. So the first thing the authors mention is that in the central nervous system, astrocytic ABCA1, meaning this protein produced by astrocytes, is crucial for the formation of lipidated ApoE. We had seen that going through the reviews last week. So there is this enzyme, ABCA1. It's an interesting protein because in addition to being an enzyme, it's also a transporter across membranes. But it has a lipidating catalytic function that can add lipid molecules to ApoE, turning it into lipidated ApoE. And in the little network that we started building last week, we had ABCA1 inducing the formation of lipidated ApoE, and therefore inhibiting the levels of non-lipidated ApoE. And the reason I bring that up right now is because towards the end of today, we may have to revise that idea a little bit. Anyway, so in the intro to the paper, the authors say that ABCA1 is crucial for the formation of lipidated ApoE, and that ApoE is an HDL-like particle that, like HDL, can take cholesterol and other lipids to neurons for the maintenance of synaptic integrity and neuronal function. In fact, they mentioned that the loss of brain ABCA1 in mice leads to disrupted synapses and causes pro-inflammatory phenotype. All of that is to say that ABCA1 is a good thing for brains, right? When you have a functioning ABCA1, you will be shuttling cholesterol and other important lipids into neurons, such that those neurons can use them for good things, for maintenance of synapses and synaptic function. When we discussed the reviews last week, I had started getting the impression that ABCA1 was not a great thing because it was creating lipidated ApoE, which would in turn facilitate the oligomerization of the amyloid beta peptides and further the formation of the plaques. But according to the intro in this paper, allegedly, one might say, well, it is normal ABCA1 function that is good for neurons. And ABCA1 turns into a bad thing for neurons and brains when perhaps becomes a little too much, too active, too expressed at too high levels, right? But under relatively normal homeostatic conditions, ABCA1 is a good thing for brains. The lack of ABCA1 exacerbates amyloid beta deposition. And I have that in red, bolding in red, because it was sort of the opposite of what we discussed last week, based on those reviews, at least. What this intro is saying is when you get rid of ABCA1, now you're actually inducing the deposition or exacerbating the deposition of A beta plaques, which is sort of counterintuitive based on what we were saying before. So last week we had it at ABCA1 through the lipidation of ApoE helps with amyloid beta deposition. This is saying that when you don't have ABCA1 is when you exacerbate amyloid beta deposition. In conversely, the overexpression of ABCA1 in a mouse model of Alzheimer's disease significantly increased circulating levels of lipidated ApoE, so far so good, in the cerebrospinal fluid and reduced amyloid plaque deposition. So what the review seemed to say last week was that ABCA1 induced lipidation of ApoE and therefore accumulation of the amyloid beta oligomers. What this introduction is saying is that somehow lipidated ApoE inhibits the formation of those A beta oligomers. So one of my goals for today is to try and tease that out and figure out why that contradiction there. And obviously, the authors of this paper, when they wrote their paper, they didn't know anything about our intellectual struggles with all this. They have their story in their mind. And in their mind, it makes perfect sense that if you were to somehow induce the activity or expression of ABCA1 in mice, particularly in mice that are prone to developing Alzheimer's disease, that would be a good thing, right? So the beneficial effects of ABCA1 in these mice are considered to be largely mediated by promoting ApoE lipidation, which we have agreed on so far, which in turn is thought to facilitate the amyloid beta peptide uptake and degradation by microglia, right? So at this point, we have some sort of a contradiction between what some of the reviews seem to be saying last week and what the intro to this paper is saying this week. But let's say, for argument's sake, that ABCA1 expression is beneficial. So I'd like to start this conversation with you guys today. How can we force brains to make more ABCA1? If you had to force a brain to make more ABCA1, how would you do that? Jacob? Oh, interesting. But where did you get that? So we saw that ABCA1 lipidates ApoE. What you're saying is if we had more ApoE in the brain, that would somehow wake up ABCA1 and wake up the brain to produce more? Huh, interesting. So you're assuming / hypothesizing that there is some sort of feedback mechanism by which more ApoE that is not lipidated would say to the brain, hey, there's a lot more of us now, we need to be lipidated, please make more ABCA1. Okay, interesting idea.
I guess like targeting the inhibitors associated with the regulation of ABCA1.
What inhibitors? If there is any inhibitors. So the idea here, Josh's idea is let's target the inhibitors of ABC1. So let's kill those. If we kill the inhibitors, we will induce expression / activity of ABCA1. Great idea, as long as we know what those inhibitors might be. Jessica?
I was going to say the same thing, but specifically one of the people mentioned
And yeah, which is the one that, so inhibiting the inhibitors is what this paper will do, right? Any other ideas that you can think of? Yeah. The what?
Like the levels of how many beta oligomers have to. It's like if there's a lot of beta.
If there's a lot of A beta oligomers, what would you?
Lower the expression of the ABCA gene.
So you're saying that a lot of A beta oligomers will somehow lower the expression of B. So how would you use that in order to try and induce the expression of ABCA1? Let's say that that is true, and this is not far from Jacob's idea that maybe there is some sort of feedback loop. Jacob was proposing from ApoE, you're proposing from A beta. If that were true, how would you use that knowledge to try and boost the activity of ABCA1? Probably lower the levels of this. Okay, so again, we'll go back to this idea of inhibiting an inhibitor. In this case, you're making A beta the inhibitor. Okay. Still hypothetical, but conceptually.
Did someone say increase the ABCA?
Hmm?
Like if we increase, did someone say increase?
No one said that. So just increase ABCA1. How would you increase ABCA1? Okay, but let's keep it less restricted to the idea of increasing ABCA1. Did you have any comments about that, Melita?
Yes, like using agonists, I don't know how to, but like,
Yeah.
to turn it off and then produce more, to increase the activity.
Turn it off or on?
Oh, sorry.
On, agonist. So, show of hands, we're all familiar with the term agonist? Kind of? Are we familiar with the term antagonist? Much more, right? So agonist is the opposite of antagonist. So antagonist is a term that is very common in pharmacology and molecular sciences, and that is any molecule that will oppose the function of your target. Right? So that's how we have antagonists of, let's say, enzymes. Agonists are any molecules that will facilitate, turn on your target, right? So what you're saying is some agonist of ABCA1. Excellent. So that will increase the function of the enzyme. Not so much the levels of the enzyme, but the function of the enzyme.
Can you make the chromatin insert where ABCA1 is?
All right. So what you're saying is somehow modify the chromatin around the ABCA1 gene such that now it becomes a little more accessible. Right? So I will take a step back, and this ties to what you were saying. Melita is suggesting to use some sort of drug to induce the activity of ABCA1. You had somewhat suggested why not somehow induce the expression. That's an idea. So maybe we can mess with the chromatin around the ABCA1 gene and induce the expression of the gene.
We can for increasing ABC, we can inhibit the inhibitors and then we can increase the agonist.
The agonist would be more or less what Melita mentioned, and that refer to activity of the enzyme once it has been expressed. Now, just like we say, maybe we can inhibit the inhibitors, and a follow-up of sorts of what Arshia was mentioning, maybe we can somehow modify chromatin. What is it that we could do to simply increase the levels of transcription of this thing, other than epigenetic modifications?
I mean, can we do it together, like agonist and inhibitor, decreasing the inhibitor and increasing the agonist?
Some sort of combination? Okay. Were you gonna add something? No?
I was going to say acetylation.
Acetylation of, okay, so if you acetylate the chromatin around this gene, and I'm guessing you're saying acetylation of the histones, you would loosen up the chromatin, making it more accessible, making it more prone to transcription, correct? The issue with that approach, conceptually a very valid approach, the issue with that approach is how do you find or how do you achieve specificity? How do you make sure that that acetylation that you're trying to force in that DNA is restricted to that gene and not many other genes that can start doing some damage, right? So the idea of epigenetic modifications to control gene expression from a therapeutic perspective is very attractive, but at the same time it always faces this massive challenge of specificity, right? How do you say, well, I'm going to throw in a bunch of histone acetylases because that's going to loosen the locus for ABCA1. Well, how do you make sure that it's restricted to that gene and not many others? Okay? So let me see. I think, do I have it here? No. All right. So let me tie some of these ideas back, and the one that ended up being a little loose. In the intro, the authors mentioned that thus far, the attempts that have been made to induce the expression of ABCA1 have to do with using agonists, but not of ABCA1, instead agonists of the transcriptional activators that control the expression of ABCA1. Remember how during the introductory weeks we mentioned that transcriptional activators and repressors can be controlled by allosteric molecules that can bind to them, change their shape, and make them bind DNA better or worse? It turns out that there is a transcriptional activator of ABCA1 called LXR. And LXR is one of those hormone receptors that will, I don't know if you heard of them before and if you remember, but hormone receptors are proteins that can bind to DNA and activate quite strongly the expression of genes, but only when they're bound to certain hormones. So that's how estrogen and testosterone and progesterone work, right? They bind to proteins that then in turn go to DNA and turn on the expression of genes. LXR is one of these hormone receptors, and LXR happens to control the expression of ABCA1. And LXR, like so many other receptors, you can hit them with agonists, with molecules that will make them change shape, go to the ABCA1 promoter, and turn on the expression of ABCA1. So that's how it had been done until this paper, and a couple papers before, simply by cranking up the expression of ABCA1 using these LXR agonists. The problem with that approach, we will revisit this idea, I think, in the discussion, is that LXR turns on the expression of many other things. And when you keep LXR active for too long, that starts becoming toxic. So even though the idea of inducing the expression of ABCA1 in brains, and the whole body, by the way, because ABCA1 also controls cholesterol metabolism. So it's really good in the context of cardiovascular disease and whatnot. So even though the idea of keeping high ABCA1 expression high is good as an idea, the approach that people knew of so far ended up being a little toxic in the long run. So here's where these people come in and say, let's look at alternatives. And they started visiting this idea of inhibiting the inhibitor, right? So they decide to inhibit the inhibitor, and it turns out that there are several microRNAs, miRNAs, that have been identified to mediate the post-transcriptional regulation of ABCA1. One of them is microRNA 33. MicroRNA 33 is encoded within the intron 16 of this other gene called SREBP2. That is a key transcriptional modulator of cellular cholesterol homeostasis. And it goes both ways. It turns out that ABCA1 is controlled by many microRNAs, and microRNA 33 controls many genes related to cholesterol homeostasis. ABCA1 is one of them, SRABP is another one, and then there is 4 or 5 others mentioned in the paper. I don't want to bog it down with the details. And here I thought we could discuss this other interesting question that is kind of only tangentially related to this paper. We have this microRNA that targets many genes that are important for cholesterol metabolism. And this microRNA itself is coded within the intron of one of the genes that it targets. So what we're talking about here is there is this gene SREBP2. We're not too concerned with that gene right now. Has a whole bunch of exons. apparently has at least 15 exons or 16, because in intron 16 there is actually the gene for, I'm going to put it here upside down. I don't know if this is the case, that it is upside down, but I'll just make it upside down, doesn't matter. That is within the intron of one of the genes that the microRNA actually controls. So, and one thing that the intro doesn't say, but I bring up here, is that this is kind of common, that microRNAs will be encoded within introns of genes that they control. So my question to you is, why do you think that species would encode microRNAs within the introns of some of the targets that they themselves control?
Well, I think if you have it as an intron, then that means if the mRNA is being processed and spliced, then you're going to have it pop out as an intron and it can go and inhibit other genes being made.
Oh, okay. So what you're saying is that these microRNA 33 won't be transcribed on its own, but that instead will be produced when this intron is spliced out, and that's when it would get processed. So your suggestion is we're going to have an SRBP, EBP, to pre-mRNA somewhere, so there is all of the introns there. Somewhere there is this intron that gets spliced out, forms that lariat, and that's when this gets processed into MI33. That's your idea?
I was just gonna say to create like a negative feedback, to help ensure like a great, like a good homeostasis between collection.
Beautiful, beautiful. Mireya, right? Yeah. Beautiful. So just to rephrase that, what Mireya is saying is, I'm paraphrasing here, I think I sort of read between the lines of what you're saying. What you're saying is the more this SRABP gene is expressed, somehow the more this gene will be expressed. And if this gene controls SRABP, it's like a built-in feedback mechanism, right? It's a built-in feedback mechanism. The more the target primer, the target mRNA is being expressed, somehow the more the microRNA is expressed that targets the host mRNA, and therefore creates a built-in negative feedback loop. I like the idea a lot. So what we're left with, though, is whether or not it is, as Sebastian was suggesting, as simple as this intron gets spliced out, and now the mRNA, the mature mRNA, is being produced. Jessica, you had your hand up?
Oh, I was just gonna say it also causing faster regulation because it's like turned on at the same time, you don't delay in between.
So it's
Like with the
close, like it's near. Along the same ideas, right? So they all sort of feed back into this idea that there's going to be a or a simultaneous production of the microRNA and the mRNA that is supposed to target, right? Conceptually, the idea is perfect. There is a technical caveat, though. You guys remember what I mentioned about when introns get spliced out, what they form? Do you guys, I kind of drew it there.
The risk.
Well, the risk complex is what will use the microRNA, but when introns get spliced out by the spliceosome, there is this funny structure that forms that we call the lariat, right? It's a little lasso that forms, and that is very quickly degraded by cells. So there wouldn't really be a chance for this microRNA to be properly processed into a mature microRNA if it is embedded within a lariat. When we say that these microRNAs live within introns, and I notice maybe my cartoon wasn't the best, but what I meant by this bent arrow here is these genes have their own promoters. They have their own little core promoters. They're not the core promoters that you would expect in a protein coding gene with TATA boxes and CG rich boxes and whatnot, because this is not a protein coding gene. It's one of those small non-coding RNAs. But these small non-coding RNAs, nonetheless, have their own little core promoters, their own little regulatory promoters that may tell them when to transcribe, and then, yes, form the hairpin that will be processed by dicer to turn this hairpin into a single-stranded microRNA that gets loaded onto the RISC complex that will go around patrolling in search of target mRNAs. Right? So, yes.
How does it know when to get transcribed? Is it like the buildup of the gene acts as like an activator towards the promoter for the miRNA?
That's right. So that's what closes the loop in a way, right? So what we're saying is this gene, SREBP and ABCA1 and all of the other targets of miRNA will have their own instructions for when to turn on, right? So there will be a series of inputs here that will turn them on. There's going to be most likely a separate set of inputs that come here to turn the microRNA33 on. How do we connect? The question becomes, how do we connect these inputs to these inputs here, right? And that is typically through metabolism. So there may be signals that turn on the expression of SREBP2 and ABCA1 and will, for instance, say, you know what, there shouldn't be any cholesterol floating around outside of cells. Let's bring that cholesterol in. And these things will do their job, will bring the cholesterol in, and when a little too much cholesterol builds up inside of cells, that may tell microRNA33, whoa, we're bringing in too much cholesterol, so why don't you turn on so that you can go target SREBP and ABCA1, tell them to calm down a little. And that's the feedback mechanism. Yeah? So one last thing that I wanted to, because I like the idea of the intron getting spliced out. And my original question was, why would these things often be encoded within the introns of the same genes they target? Your idea was really good in that sense. It's simple. You express the target gene, you're expressing the inhibitor. Simultaneously, it's a built-in negative feedback loop that doesn't require any further inputs. Then I come in, party pooper, and I say, no, that won't work because the lariats get destroyed quickly. So you need some sort of independent input. Except that one of the reasons why, evolutionarily speaking, these things are often, the terminology we use is nested in introns, is because when you start transcribing the target gene, in this case the SREBP gene, what do you need to do to that region or that area of the chromosome? You need to completely unravel it, right? So you need to make a CRC, a chromatin remodeling complex, go through that region and take out all the nucleosomes. What do you think that will do to this nested gene here? Immediately it makes it way more available, right? So it's almost like the cell is saying, I will express this SREBP2 gene, and in the same action, I will make the negative feedback inhibitor accessible. So I'm gonna, in a way, be ready to listen to those signals that say, slow down a bit with this SREBP. So I'm expressing SREBP, and I'm preparing myself to be very attentive to signals that say, stop now. That's one reason. Another argument that is made often is when in a genome you have all genes completely scattered around, any mutation that falls on any one of them could take them off, right? So what happens is if there is a bunch of genes that need to somehow act in concert, that they make sense only in the company of others, if you mutate any one of them, you mutated only one, but you may have messed things up for every other one. In this particular case, let's say that miR33 lives somewhere else in the genome. And just by bad luck, it gets hit with some sort of mutagen and it acquires a mutation. So now SREBP is completely out of control, right? And that, evolutionarily speaking, may not be a good thing. So what evolution often does is it puts the genes that work together, it places them together in the genome, such that if there is some sort of mutation that breaks SREBP, it may also be breaking miR33. So now you don't have an imbalance of genetic function. Either both stay or both go. And that, the argument goes, it can be better for genetic regulation because it creates for a much quicker and stricter selection against mutations. It's one thing to lose miR33 function, but keep an unregulated SREBP. That may be a little harder for evolution to figure out what to do with this. The other way around as well. What if we lose SREBP, but we maintain miR33 function? I don't know what to do with this. But if I lose them both, then I know to select against these organisms much more quickly. So often what happens is evolution will place these things together. One way to think about it is such that if one goes, both go. Another way to think about it is if evolution will protect this area because the SREBP gene is there, it will also protect miR33. So it is protecting against mutations that affect the actual gene with a function, SREBP, and also its regulators. Did that make sense? So that's another evolutionary argument for why microRNAs are often nested within the sequences of the very targets they target. And if biology taught us anything, is that it's probably a little bit of both. Okay? We're good? All right. So it turns out that in going with this idea that adding too much of the LXR agonist to induce expression of ABCA1 was in the long run toxic, people started experimenting with the idea of administering miR33 antisense oligonucleotides, so things that would break miR33, in going with this idea that if miR33 is an inhibitor of ABCA1 and I can somehow inhibit the inhibitor, I will indirectly be inducing what I want, ABCA1 expression. And previous work has shown that administration of these miR33 ASOs or antisense oligos in mice and non-human primates increases ABCA1 expression, might say as expected, enhances plasma HDL levels, which promotes reverse cholesterol transport, so cholesterol back into cells and not floating around in the blood, and retards the progression of atherosclerotic lesions. So what this research group wanted to see is if this approach would work in the brain. Okay. All right. So first experiment they do, they take neurons astrocytes and microglia. If you guys recall, neurons, the ones making electrical connections and carrying information in the brain. Astrocytes and microglia, two types of glial cells that support neurons inside of brains. So they take these cells in culture, and then they expose them to a miR33 antisense oligonucleotide. You can think of it as one of those siRNAs that we mentioned a couple of weeks ago. So it's a short RNA molecule with a sequence that is antisense to what you want to inhibit or controls. And what they find is, so the cells treated with these miR33 antisense, what they did is measure the levels of miR33 in these cells, and as they add more and more of these oligos, as the concentration of the oligos increase in all three cell types, the less and less you have of miR33. So incubating cells with these miR33 antisense is effectively reducing the levels of miR33 in these cells. Okay? Those are the black bars. So far, so good? The white bars are controls, and they're there to show that when you don't do anything to these cells, the levels of miR33 stay steady. My question to you is, what do you think is a good control in some of these experiments in which you use antisense oligonucleotides or these siRNAs? What would you do as a control? CD?
Maybe a non-targeting ASC, which does not target MIR33, like which does not bind to MIR33.
Excellent. Yes, it's very common to think, well, a control is just cells that you don't add anything to. You just add water, right? That's the control. You just don't do anything to them. But in these experiments, and I show that in the next slide, the control is actually a control antisense oligonucleotide. It is, as Siri was saying, an oligonucleotide that won't target miR33. In fact, as far as you can tell, it won't target anything. But you still want to have an oligonucleotide there. Why? Because when you introduce oligonucleotides in cells and they have a distinct target molecule inside of those cells, they will go find them, and in this case, they will attack the miR33 and block it somehow. But that is not to say that that's the only thing that these oligonucleotides will do to those cells. Maybe, and in fact what has been observed over and over, is that the mere presence of these oligonucleotides can trigger a reaction in the cells. Cells don't like to have these things around that are not 5' capped, they're not polyadenylated, they're short RNAs. They don't recognize them as a legit microRNA or something, or a small nucleolar RNA, something they produce. So what happens is sometimes these molecular species in large quantities inside of cells will trigger biochemical reactions on the part of the cell, right? So whenever you're doing experiments with antisense oligonucleotides, the proper control is to have another oligonucleotide that, as far as you can tell, won't target anything on the cells. It's almost like shooting blanks of sorts, right? So it's you have all of the biochemical properties of an oligonucleotide without any known biological target, so that you can tell if what you are measuring may be due to the inhibition of your target or may be due to these more generic reactions from the cells. Right? This concept of, in my control, I'm going to do everything I did in my experimental minus that last step, applies across the board. If I ask you, for instance, I'd like to investigate the effect of injecting mice with Gatorade and on heart rate, right? What would be your control? Many of you may think, well, just a mouse without any injections. But then the counter argument is, well, let's say that I inject the Gatorade and we see heart rate go up. How do I know that it was the Gatorade that I injected or the stress of the injection? Maybe that injection is stressful enough that poor little mouse, heart rate went up. So what do? Our control should be an injection of saline or water or preferably saline, right? So it's always the same. I don't know, have you ever heard of sham operations? No? Sham operations, yeah. What that is whenever you're doing an experimental surgery in an experimental organism, let's say, once again, a rat this time. You're doing an experimental surgery in a rat. You're taking out part of the liver, for instance. Your control shouldn't be a rat that you don't touch. It is a rat that, and I kid you not, a sham operation is you grab the rat, anesthetize it, cut it open, expose the liver, and just as you're about to cut a portion of the liver, you don't. And then you put everything back in, sew it up. That's your sham operation. So you did everything that you would do in your experimental except that last bit. Same here. A control ASO is an antisense oligonucleotide, antisense in that it is an oligonucleotide that as far as you can tell is not going to be antisense to anything. But the cells go through the same feelings of getting exposed to these molecules in culture. Okay? Yeah? Cool. So, treatment with these miR33 oligonucleotide or antisense decrease the levels of mature miR33 in a way as expected. And what they saw is, in a way as well as expected, expression of ABCA1 increased. So what you see in these three panels are Western blots. So we're not looking at levels of ABCA1 RNA or mRNA expression here. We're actually looking at the actual protein. And the darker these bands, if you're not familiar with Western blotting, the darker the bands, the more of the protein there was in your sample. And in all three cases, I lost the labels here, but these are neurons, astrocytes, and microglia. The more of these antisense against miR33, the more ABCA1 these cells express or have in them. Okay? So see, I just made that mistake that I was expecting to or hoping to discuss with you. So what these blots tell us is that there is more ABCA1 in the lanes corresponding to the samples treated with the anti-miR33. One might say that there was indeed more ABCA1 in those samples, and that's why those bands are darker, because those samples they had more ABCA1 in them. But why else could there be more ABCA1 in those samples? What kind of experimental error can one make that now you see more of this protein in your sample? Does the question make sense?
Like, how, like, okay, if you're saying why there are different, like maybe the way they transferred it?
Transferred from where?
Like, is that what you're trying to say? Like, if there was, why was there a mistake?
Well, what I'm saying is that once one looks at these darker bands and one might conclude, oh, these samples they had more ABCA1 in them. But what alternative explanation could there be for there being more ABCA1 protein present in those places?
Loading control?
Oh, loading control. What does that mean?
Like the way they transferred it? Like maybe they
Transfer from your sample to the, exactly. So what happens is, and you'll see if you hadn't come across these type of experiments before, I'll show you something that you'll see all of the time. And that is the concept of a loading control. The idea here is, this sample here that I treated with 1 micromolar of the antisense has, 5 more times ABCA1 than my control. I can come in with my professorial voice and say, those cells had 5 more times ABCA1 in them. But a devil's advocate might say, no, maybe you made a mistake and you loaded 5 times more sample in that well. Maybe just a human error, you loaded way more total protein in those wells. No wonder that you're going to see more of ABCA1. You loaded more of everything. Does that make sense? Yeah? So that's where the loading control concept comes in. And it goes always like this. You will look at your protein of interest, in this case ABCA1. This is the one that we want to learn more about or want to find out about. But then you will use something called a housekeeping gene. A housekeeping gene is any gene that is thought, at least, to be expressed by cells at very similar levels no matter what. We call those housekeeping genes because they play often very central functions for the cells, so they're not regulated. Cells never go through some sort of thinking process about whether or not they should express housekeeping genes. They need them, no matter what. Nourished or starving, hot or cold, day or night, young or old, cells will always need their housekeeping genes, and they always express them at relatively steady state levels. So anytime that you see a Western blot, you will always see, or almost always see, some sort of housekeeping gene. In this case, they use tubulin. It's one of the common ones. And the idea here is, if I see a lot more ABCA1, but similar levels of tubulin, it means that I didn't mess up in my loading of the samples. If, on the other hand, my ABCA1, let me do this real quick. Come on. If my ABCA1 was darker, as shown here, but my tubulin is really darker as well, then I can start making the argument, well, maybe I messed up and I added a lot more of my protein mix to this experiment. And that's why I'm seeing more ABCA1, because I had a lot more protein in total. Right? So you will see in papers that often any Western blot against proteins of interest are accompanied by Western blots against housekeeping genes that serve as loading controls. They control for equal loading across the groups. So in this case, the equal loading of tubulin says that we can pay attention to the differences in the amounts of ABCA1. And in this paper and in many others, you will see often these images of the blots along with densitometric analysis. What you do is you do these several times, and each of those times you ask a computer to measure the darkness, if these bands quantify it, turn it into numbers, such that from each of these replicates of your experiment, you can generate numbers that you can plot with bars. So in this particular case, these are bar graphs that show the relative ratios of ABCA1 to tubulin, right? So tubulin stays the same, ABCA1 goes up. That means that the ratio of ABCA1 to tubulin will go up as well. And we see that happening in neurons, astrocytes, and in microglia. Yeah, Gabi?
I'm confused about how the ABCA1 can look darker on the bar if you're inputting the same volume all throughout, just different.
Because what you're inputting the same all throughout is total protein present in the cell. Now, it turns out that that total protein mix has proteins that may be differing in relative amounts. So I don't know exactly what went into this particular experiment. For argument's sake, let's say that as the experimenter, I decide that I will load 10 micrograms of total protein in each of these lanes. Those 10 micrograms I measure separately, and I know that I'm scooping 10 micrograms and putting into the well. Within those 10 micrograms, there may be a lot more of ABCA1, which may mean that there is a lot less of something else, or that by scooping a total of 10 micrograms, I may be leaving behind other things. Does that make sense? Let me see if we can come up with this analogy. Let's say that you go to a market, right? And let's say better. You go to a farm, right? And you're gonna go get some eggs and produce. Turns out that your bags will hold only a pound of stuff, right? If there is a farm that produces a lot more eggs, you may fill up the bag with eggs, but that means that you're gonna get to that pound of stuff, leaving some tomatoes behind. Does that start making sense? So if the actual cells are expressing a lot more ABCA1, you're gonna see a lot more of ABCA1, even though the total amount of protein is the same that you loaded in those. Yeah? Now, why isn't that argument applying to tubulin? Because tubulin, being a housekeeping gene, is always expressed at high levels than any other thing. Yeah? That makes sense for everybody? Cells are mixtures of things, right? And when you're scooping a total microgram of everything, if you had a lot more of ABCA1, you will reach that microgram sooner, having grabbed more ABCA1s and leaving behind other things. Yeah? All right? Okay. Trying to think, we may have to actually leave some of the rest of the discussion for Wednesday and couple it with the next paper. But let's finish with this thought at least. So the conclusion from this experiment, the straightforward conclusion from this experiment, is that the miR33 antisense oligos lead to increased ABCA1 expression, right? We have our loaded controls, we did the densitometry. We know that the more of the antisense against miR33 we add, the more these cells have of ABCA1. We think that it's because the ABCA1 mRNAs are being de-repressed. Right? What we knew before going into these experiments is that miR33 is a microRNA that normally targets ABCA1 mRNAs. So if miR33 inhibits ABCA1 mRNA translation or induces their degradation, if we inhibit the levels of miR33, then we should expect to see more ABCA1 protein in those cells. So far, so good. But how else could the levels of ABCA1 protein increase within cells? If you have cells and you see more of the protein, one explanation is that the mRNA that was present is being translated more. I agree with that. But as scientists, we always play devil's advocate. We have to be our strongest critics of sorts, right? So whenever we see that the levels of ABCA1 protein are going up, we think that it's because the mRNAs for that protein are being translated better. But what other alternative explanations could there be?
And the proteins itself, they don't die.
Excellent. So less protein degradation. Love it. Less ABC1 degradation. I like it. What else? Maybe people who didn't participate yet today? how else could you crank up the levels of ABCA1 protein other than de-repressing translation or inhibiting their degradation? All right, anyone?
Maybe like an activator and an enhancer.
Activator or an enhancer of what?
ABCA1.
Of the ABCA1 what? The protein or
Yes, just the expression.
expression of the protein? So remember, guys, when we talk about expression of the protein, it all starts with transcription, right? So some sort of activation of transcription. Is that what you had in mind, more or less? Yeah. Whenever we see more of a protein in cells, maybe there's just more of the mRNA for it, right? So it could be less protein degradation, or it could be more ABCA1 mRNA. And how could there be more ABCA1 mRNA in cells? You touched on one, which is just transcribe it more. How else could you have more mRNA in those cells?
How we talked about last week, how there's a lot of mRNA, but they're not being translated yet.
Uh-huh.
So something like that, when the cell needs it to be translated, they'll allow for that.
So increased translation of the mRNA. Okay. Increase mRNA translation. All right. 1, 2, all of these ideas. Any other thoughts? Yeah. Less mRNA. So the more mRNA we can achieve with more transcription or less degradation. Sorry, the writing is getting a little mangled there, but yeah, or less mRNA degradation. Correct, right? Which is, in a way, one of the things that you might expect by inhibiting miR33, because miR33, being a microRNA, it will for sure inhibit the translation of its target mRNAs, and in some cases, it may induce its degradation, right? Do you remember when it was either case? When was it that it would just inhibit translation versus induce degradation? What determined that?
Like if the base pairs were imperfect, that would inhibit it. And if they were perfect, that would
Destroy, right? So it could be that miR33 has a perfect match in ABCA1 mRNA, so it would normally destroy it, but because we're inhibiting it with the antisense, then now the mRNA is preserved. So the point is that, and this is the point that I wanted to emphasize with you guys, the authors of the paper designed the experiment such that they knew that with the anti-miR33, they expected to see more ABCA1 by design. They know that miR33 targets the ABCA1 mRNA, most likely to prevent translation, best case scenario to destroy it. If you get that miR33 out of cells using this antisense, you should see more ABCA1. That's it. They do the experiment, they see those results. Case closed. But these scientists, being good scientists, said, let's think of alternative explanations. Let's make sure that we cover all the bases. And they decided to do something that wasn't all that necessary, but they went ahead and measured the mRNA levels of key miR33 targets like ABCA1, ABCG1, cousin of this thing, HAD-HB, and CROT. And what they found is no measurable differences in transcript levels. So basically, what they're mostly addressing there is this idea that maybe there was more ABCA1 protein because there was more mRNA. What this experiment that they don't show in the main paper, they show in supplementary figures, what they're saying is, no, there was no more mRNA. There were comparable levels. We didn't see any measurable differences. But it seems like this mRNA is being translated better into protein. Okay? Actually, if you were the reviewer of the paper, you would have said, well, how do you know that those proteins are not being degraded? And then they would have had to go do an experiment to make sure that it's not less degradation of the protein.